Good morning. Today's topic is biofilm in endodontics. Myself, Dr. Saranya from the Department of Consulted Industry, and the contents that we are going to deal is regarding the definition, stages of biofilm formation, ultrastructure of biofilm, quorum sensing, types of endodontic biofilm, methods to eradicate biofilm, and conclusion. The microorganisms can survive in the root canal even in the environment of low oxygen tension. Most commonly, gram-positive facultative anaerobic bacteria in the coronal portion, followed by the gram-negative and another anaerobic bacteria. Due to the lack of blood supply and compromised host defense, makes a pulp a reservoir for the infection. The tissue fluids, disintegrated cells, necrotic tissues, and low oxygen tension determine the predominance of bacteria in the pulp chamber. The common genera of the infected root canals are mainly the obligate anaerobes and the facultative anaerobes. The obligate anaerobes include the gram-positive cocci, gram-positive wores, gram-negative cocci, gram-negative wores, and spirochetes. The commonly found gram-positive cocci are Streptococcus, Streptococcus. The gram-positive wores are Actinomyces, Lactobacillus, Eubacterium. Gram-negative cocci are Veillonella. Gram-negative wores is Prevotella, Porphyromonas, and spirochete is Treponema. The facultative anaerobes mostly include the gram-positive cocci, gram-positive wores, gram-negative cocci, gram-negative wores, and yeast. Now, the definition of biofilm. Biofilm is a mode of microbial growth where dynamic communities of the interacting sessile cells are irreversibly attached to a solid substratum as well as each other and are embedded in a self-made matrix of extracellular polymeric substances called as EPS. And the dental plaque is the typical example of a biofilm. There are many stages of this. The first stage is adsorption. The adsorption of macromolecules in the planktonic phase to the surface that leads to the formation of a conditioning film. And it is made up of mainly the proteins, glycoproteins, and DNA. This is the attachment phase. The stage 1 is called as the attachment phase. Stage 2, it is called as the growth phase. In this, the adhesion occurs. That is, the adhesion and the co-adhesion of the microorganism, and the attachment may be strengthened through a polymer protection and unfolding of cell surface structures. In this, the proliferation of the microorganisms to form microcolonies. The third stage is the maturation stage. In the third stage, that is in the maturation stage, the multiplication and metabolism of the attached microorganisms that ultimately will result in a structurally organized mixed microbial community. That is in the maturation stage, a metabolism of the attached microorganisms that is ultimately will result in a structurally organized mixed microbial community. And it takes around hours to complete the maturation phase. The last stage is the detachment phase. That is the stage 4. It is called as the detachment of the biofilm organisms. It takes around days to weeks to complete the detachment phase. So basically there are 4 stages. The detachment stage is also known as the dissemination stage. Now, moving on to the ultrastructure of the biofilm. Basically, the structure of the biofilm includes the microcolonies and the glycocalyx matrix. That is, it includes the bacterial microcolonies and a glycocalyx structure within it, the substrate and the chemical gradients in the microenvironment. Microcolonies, they are the basic structural unit of a biofilm. It is the microcolonies of the cell clusters that is formed by the surface adherent bacterial cells. Microcolonies are discrete units of densely packed bacterial cells aggregates. They can be either in a single or multi-species aggregates. glycocalyx matrix that is made up of extracellular polymeric substance, that's EPS, which surrounds the micropolymerase and anchors to the bacterial cell. A fresh biofilm matrix is made up of biopolymers such as the polysaccharides, proteins, nucleic acids, and salts. Around 85% is the matrix and 15% of the cell is the structure of the biofilm. The water channels, which are regarded as a primitive circulatory system in a biofilm. These water channels intersect the structure of the biofilm to establish the connections between the microcolonies. It includes a function such as they facilitate the efficient exchange of materials between the bacterial cell and bulk fluid. They help to coordinate functions in a biofilm community. The structural feature of a biofilm that has the highest impact in chronic bacterial infection is the tendency of the microcolonies to detach from the biofilm community. During the process of detachment, the biofilm transfers particulate constituents from the biofilm to the fluid bathing biofilm. Now comes the crowd sensing, which is frequently asked remark shortly. The quorum sensing is a term that is currently used for systems based on that cell-to-cell communication by microorganisms that regulate gene expression mediated by low molecular weight molecules. These low molecular weight molecules are called as the signal molecules. The sufficient signal molecule act as an auto-inducer to increase the expression of the specific genetic traits that would enhance the chance of survival of the groups. There are both gram-positive as well as gram-negative organisms. One of them they are the planktonic bacteria. The concentration of the chemical signals separated by the plankton cells is low. The low concentration of the signal molecules does not change the genetic expression. Biofilm bacteria, they include some biofilm cells that are held together in dense populations. So, the secreted chemical signals higher concentrations. Signal molecules then recross the cell membrane and trigger the changes within the genetic activity. There are different types of endodontic biofilms. They are the intracanal biofilms, that is, the biofilm that is present within the pulp chamber. The extraradicular biofilm, that is, the biofilm that is present on the outer surface of the root. The periapical biofilm and the biomaterial center infections. The periapical biofilms are nothing but the biofilm that is present on the periapical region, and the biomaterial centered ones are those biofilms that are surrounding the external material that is being used within the tooth surface for restoration. Intracanal biofilms that is formed on the root canal dentin. The site is the microbial biofilm that is formed on the root canal dentin of an endodontically infected tooth. The structure, the bacteria exists as loose collections and it is made up of foci, rods, and filamentous pattern. Extraradicular biofilm, they are also known as the root surface biofilm. They are attached at the center. Teeth with asymptomatic periapical periodontitis, chronic periapical abscess associated with the sinus tract are the sites of the extraradicular biofilms. The structure is basically dominated by the foci and the short rods. Periapical biofilms are mostly present in the periapical region of an endodontically infected teeth. They are dominated by the presence of actinomyces and propionine bacterium. Biomaterial centered infections. These are site that is present in the biofilm surrounding the gutta percha and cilius. The microorganisms basically present in a biomaterial centered infection are Streptococcus aureus, E. faecalis, and gingivalis. Then Now comes the battery biofilms. The method to eradicate biofilms. Let's see what are the methods that can be done to remove the biofilms. Biofilm is basically present within a type of biofilm that is present within the canal is the intra-radicular biofilm. And in order to eliminate that, during the root canal procedure, we can use various irrigants such as sodium hypochlorite, chlorhexidine gluconate, cumix, iodine, UTA, MTAD, tetraquine, calcium hydroxide, and the various agitation techniques such as ultrasonically activated irrigation, ozonated water, lasers, plasma dental probe, photoactivated disinfection, antibacterial nanoparticles, and an endoactivator system. By using these irrigants and irrigant activation systems, the biofilm can be eliminated from within the root canal. The effect of irrigative solutions on the root canal biofilms. While none of the irrigating solutions, disinfecting agents presently used in the neurotic treatment are able to do all the required tasks, a combination of the solutions might be the key. The effect of irrigating solution or the root canal. The action of the sodium hypochlorite is the peptide rings of the bacteria are broken up and bacterial proteins are dissolved. In water, the hypochlorite ion ionizes into the sodium ion and the hypochlorite ion. Hypochlorous acid is responsible for the antibacterial activity. Chlorhexidine, due to its cationic nature, it is capable of electrostatically binding to the negatively charged surface of the bacteria, damaging the outer layers of the cell wall and rendering it permeable. Depending on its concentration, it can have both bacteriostatic and bactericidal effects. EDTA is often suggested as an irrigation solution because it can chelate and remove the mineralized portion of the small layers. EDTA extracts and chelates with bacterial surface proteins by combining it with ions from the cell envelope, which can eventually lead to bacterial death. Cumix that consists of EDTA, chlorhexidine, and detergent. That's cumix is a mixture of EDTA, chlorhexidine, and detergent. It has effective as 6% of hypochlorite in killing 1-day-old e-PADASHALA, but slightly less effective against the bacteria in 3-week-old biofilm. Local intracanal medicaments that should not easily replace by the tissue fluid and that can remain physically intact over weeks or months. Basically, calcium hydroxide is strongly alkaline in nature and it dissociates into calcium and hydroxide ions in aqueous solution. The latter provide antimicrobial effects and tissue dissolving capacity. Its most essential function is then to obstruct the regrowth of the bacteria within the canal. Now, moving on to the recent advances in combating the biofilm, photon-activated disinfection, antibacterial nanoparticles, endoactivator system, ultrasonically activated irrigation, lasers, and ozonated water. Going on to the conclusion, biofilms are everywhere and can develop on virtually every natural and man-made surface. Biofilm formation has been demonstrated for numerous pathogens and is clearly one of the main strategies for bacterial survival in a variety of sites within the human body. Bacterial biofilms are very prevalent in the apical root canals of the teeth with primary and post-treatment apical periodontitis. While none of the mechanical and the chemical factors alone can predictably eradicate the effective agents, their combined action under optimal efforts is a key factor for long-term success in the endodontic treatment. These are the important questions or the frequently asked questions that's being asked for the university exam. Others. Definition of biofilm and the different stages of biofilm formation, the structure of the biofilm.